Let's remember what we've learned about the difference between covalent and non-covalent bonds. Covalent bonds are strong. It takes a couple of hundred kilojoules per mole to break one, and they involve the sharing of electron pairs in the valence shell of atoms. Non-covalent bonds are much weaker, and they don't involve electron sharing. Most are based on attractive forces between polar or ionic molecules, and the attraction can be within one molecule or between two molecules, that is, intramolecular or intermolecular. We've already talked about hydrogen bonds, a form of dipole-dipole interactions that is extremely important in biochemistry. In this video, we'll talk about electrostatic interactions, van der Waals forces, and hydrophobic interactions. Biomolecules are sometimes not just polar, but fully ionized. Electrostatic interactions between molecules or groups with opposite charge can be quite strong, potentially the strongest non-covalent interactions. These charge-charge interactions also have a longer reach and don't depend on molecular geometry in the way that hydrogen bonds do. The most relevant example for us is the interaction between acidic and basic amino acids in proteins. For example, here between glutamate on the left and lysine on the right. These interactions are sometimes called salt bridges, or more accurately, ion pairs. If they are on the surface of proteins, the interaction is sometimes weakened by shielding or screening effect from water molecules that arrange themselves around the ionized groups, and ions in solution can also screen those charged groups. If the salt bridge or ion pair is buried deep within the protein, there is no such screening effect, and in that case, the bridge is much stronger than between solvent-exposed ion pairs on the surface of proteins. Let's now look at the third type of non-covalent interactions, the van der Waals forces. These are a bit trickier to understand. Van der Waals forces act between dipoles that can either be permanent or inducible. For example, if two carbonyl groups are stacked head to tail in just the right orientation and at the right distance, there are attractive van der Waals forces of about 10 kilojoules per mole between them, so about a third of the strength of hydrogen bonds. The interaction between inducible dipoles is a little trickier to understand. So here's the thing. Even completely non-polar bonds, like the carbon-hydrogen bond here, are sometimes very temporarily polarized because of just random asymmetries in the distribution of electrons around the nuclei. If there's an asymmetry in one place, it has a knock-on polarizing effect on its neighbor. We have to imagine this as quite a fast flickering back and forth. The interaction between these transient dipoles results in an attractive force between really any two atoms, but it does depend strongly on the distance between them. So this force is really only strong if the atoms are very close, but once they get even closer, there is a repulsive force, because the negatively charged electron clouds of the two atoms repel each other. Overall, this results in a curve like this, with a trough where the van der Waals attraction is greatest. The distance between the two atoms at this point is the sum of their so-called van der Waals radii, and that is in the range of 0.1 to 0.2 nanometers per atom. To be sure, the attractive force at this optimal distance is very small. It's less than 1 kilojoule per mole. But strength comes in numbers. Whenever clusters of atoms are tightly packed in biomolecules, a large number of these van der Waals interacting forces stabilize the molecule. That is, for example, the case in DNA. The stacked base pairs are, among other things, held together by van der Waals forces. So let's summarize. Van der Waals forces act between stable dipoles in polar molecules or inducible dipoles in any kind of molecule. Inducible dipoles come about by random asymmetries or fluctuations of electron distribution. These forces are strongest at very close range and are 1 or 2 orders of magnitude weaker than other non-covalent interactions, but these interactions add up to quite a significant contributing factor to macromolecular structures. Finally, hydrophobic interactions. These are not bonds, and crucially, they are not based on attractive forces between molecules in the way one would think. Hydrophobic molecules have few or no polar bonds, and that means they cannot engage in hydrogen bonds with water molecules around them, which means they are poorly soluble. When hydrophobic molecules are dissolved, they force the water molecules around them into a very specific arrangement. The bulk water, in contrast, is free to wiggle around and form hydrogen bonds in all directions. By clustering the hydrophobic molecules, fewer water molecules are forced into this more ordered arrangement. What makes the situation on the right more favorable is therefore not the energy released through bond formation, but the increase in disorder or entropy, delta S. This is how you can calculate the effect. For example, for every CH2 group of a hydrocarbon that is moved out of the water, around 3 kilojoules per mole are gained in entropy, so this is a favorable process. You don't have to pull them out of water, though. It's enough to screen the hydrophobic groups from interactions with water. And so huddling together with other hydrophobic molecules does the same trick. So let's recap. There are 4 main types of non-covalent interactions that are relevant for biochemistry. Hydrogen bonds are a particular type of dipole-dipole interactions where a hydrogen atom is sandwiched between 2 electronegative atoms and is covalently bound to one of them. Electrostatic interactions are based on the attraction of opposite charges. Technically, the repulsion of like charges is an electrostatic interaction as well. The main kind of electrostatic interactions that's interesting for us here is the salt bridge or ion pairs in proteins, and these are more stable if they are buried deep inside the structure. On the outside of the protein they are weaker because of screening effects from water and ions. Van der Waals forces act between stable or inducible dipoles. They are weak and short-ranging, but in tightly packed molecules like DNA, proteins, or lipid membranes, these forces add up to a significant factor. And finally, hydrophobic interactions are based not on attraction between hydrophobic molecules, but on the increase in entropy or disorder when water molecules are freed from the orderly arrangement that a hydrophobic molecule imposes on them. Let's compare the strength of those non-covalent interactions. If we take the carbon-hydrogen bond as a benchmark at around 430 kilojoules per mole, ion pairs or salt bridges are potentially the strongest of all non-covalent interactions, but only if the charges are not screened. Next are hydrogen bonds at 2 to 30 kilojoules per mole, and finally van der Waals forces are only about 1 kilojoule per mole per interacting pairs of atoms. But as you remember, they do add up. And finally, hydrophobic interactions are in a similar range as hydrogen bonds, but we need to remember that the principle is different and that we're not talking about an attractive force between hydrophobic molecules.